Yeah, thank you very much, Anita, for your kind words for introduction. And that gives me the chance to thank the interpreters. They do a significant and very useful job for all the foreigners who are not able to follow the Serbian presentations and discussion. Thank you very much. So I could follow also the kind words you used to introduce myself. I don't think, or I have the impression it was a little bit too much, and probably your expectation is increasing, so it's a challenge for me to meet your expectations. So thank you very much to invite me to come here. It's also my very first time in your country. I've never been to Belgrade, I've never been to this place, very nice area, and I was happy when you invited me to come. So thanks again, and thanks also for preparing the translations to make it easier for you to follow. And that is also, I think, a stage where I should express some excuse to those who prepared the translations, because they have to deal with such complicated males like myself, working on the presentations until the last minute. So I made some changes, and we had to work on that this morning. So thanks again. And now I'd like to discuss together with you the issue of bioavailability and bioequivalent studies with modified release formulation. It's very difficult to talk about this topic in only 30 minutes. We could have a conference on that, but I think that's same with Vinod's excellent presentation. By the way, I think also it was really wise of the organizers to choose this date of the month, because it gives us the chance to congratulate our American citizen, Vinod, that the American people was so kind to elect this time the European favorite candidate as president. So congratulations to that. So now to science. So it's very difficult to cover that in 30 minutes. However, I'd like to do my best. As you already heard this morning, there is a revised version of the existing European guidance, note for guidance. Guidance for industry is the way the American colleagues call their guidelines. And this is the revised version of the existing CPMP note for guidance on bioavailability and bioequivalence. And there are some significant changes. I would like to say they are paradigm shifts. First of all, it's a shift from a more general or scientific approach to stricter regulations, more cookbook concept. I'm not very happy about that, I have to clearly say, since it is my deep understanding that every single study needs a clear definition of the scientific rationale behind. And it's impossible to cover all the projects and all the problems to be investigated by the studies with one general approach. One size fits all is not possible in bioavailability and bioequivalence, and therefore the question is it possible to cover all these aspects by one regulation? But this will be discussed again since we are in the discussion process of the revised version of the guideline, and it's my pleasure to invite you for an open forum which is organized by the Network Biopharmaceutics, Bioavailability and Bioequivalency of UFAPS, the European Federation of Pharmaceutical Sciences. And this will be held in Bonn, Germany, next January. That's not the best time to come to Germany, weather-wise, but as the deadline for comments has been set to 31st of January next year, we had to organize it in this particular time frame. However, it gives you the chance just to listen to the presentations, and it's not attractive to go outside because it's cold and probably rainy or some snow. So this is your chance to contribute to the guidance process, and I really like to motivate you to come. This is organized as an open forum, so it is your time to present comments, ask questions to the regulators, and we are willing to combine and summarize it later on in our comments to the guideline. Now, what is under discussion concerning modified release formulation? So the answer is simply nothing. This is the general guideline and not the specific one for modified release products. However, there are certain aspects which are affected by the new regulations. One is, and I will go a little more in details into that, the necessity and appropriateness of multiple dose studies for modified release formulations. Are they necessary or not? If you look into the US FDA guidance for industry, it is not necessary, not mandatory for generic drug approval. In Europe, it's still in the guideline, and we will have a discussion on that. And then standardization of meals in food effect studies, and I will only briefly go into it because that is topic of the next presentation. And some of you know that it is my intention also to suggest to have some individualization because due to our experience in food effect studies, and we did a significant number of those, is that if you force a 50-kilogram young small lady to eat this American high-fat breakfast, 1,000 calories within 30 minutes, it's really, you suffer even if you look at her, and sometimes it's impossible to eat it within this time frame. On the other hand, the 200-kilogram big guy who has finished the meal within 5 minutes, and he is already hungry after 2 hours, and the stomach is empty. Housekeeping waves are occurring, while the lady is filled up for the next 24 hours. No gastric emptying occurs. So that is something which we should investigate a little more in details. However, it's not the topic of my presentation. Now, what are the requirements? And I make reference to the European CPMP Note for Guidance, which has been published in the late 1990s. And of course, the background again is that the products have to be efficacious and safe. And if you design a study, the specific characteristics of a modified release dosage form, prolonged release, enteric coated, delayed release, or whatever modified release formulations have to be considered. So it's again up to you. And I think I would like to repeat what I said this morning as a comment. I think you should make up your mind to define clearly the scientific rationale for your study. Don't believe that the guideline should do your job and covers all the questions. So the specific characteristics should be considered. What are the specific characteristics compared to immediate release formulation? Normally, there are relatively high doses in the modified release formulations. If you consider, for example, a once daily administration form, it has to administer the dose for one entire day compared to a 3 times or 4 times administered immediate release formulation. So there's a higher dose, and that's the reason why dose dumping is such an issue in the regulatory thinking. Then the controlled drug delivery, which has to be investigated, the interaction with food, which I already mentioned, and a consistent gastrointestinal transit. So the GI transit is very important in the case of modified release formulation, much more important than in the case of immediate release formulation, where drug release occurs normally inside the stomach. Here, the entire GI tract is relevant, and in particular, the absorption site, etc., etc. So this is essential. And there are 3 scenarios which are defined in the guideline, and these 3 scenarios are the modified release formulation, which is developed as the first preparation of a new molecular entity. In other words, the originator innovative drug is introduced in the market not as an immediate release formulation, but directly as a first preparation as a modified release form. Second scenario, the development of the first modified release formulation of a known drug substance. In other words, this new molecular entity has been introduced as an immediate release formulation first, and years later, the development of a modified release form is intended. And third, the generic development of a modified release forms of an innovator product which is on the market as modified release dosage form. So these are the scenarios, and the objective is that the biopharmaceutical characteristics should be investigated and characterized in vivo, and the goal is to confirm therapeutic equivalence, as Attila Hinchal very elegantly explained this morning. That is the concept, that the assessment of bioequivalence is interpreted as the proof of therapeutic equivalence. That's the concept for generic drug approval worldwide. So that's the background. So let's briefly go through these three scenarios. That's the first one. The new chemical entity is introduced as a modified release formulation, and it's clear that under these circumstances, all the essential chemical and pharmaceutical investigations have to be performed, and also the preclinical and the complete clinical package has to be submitted in the dossier to the authority. That is an innovative drug application, and this is very similar if you look into the FDA regulation, and the product has to meet the controlled release claims, that's for sure. You have to show that your modified release formulation is really a modified release dosage form. The profile should rule out dose dumping, so dose dumping has to be avoided, and the product provides consistent pharmacokinetic performance. That's a very essential point I'd like to point out again. So the product has to exhibit consistent in vivo performance. Now, what are the requirements, the study objectives for bioavailability assessment for the first modified release product? There should be a comparison of the modified release dosage form with an immediate release preparation, which is on the market as the first formulation. And the questions to be answered are, is the modified release absorption achieved, and is that shown by the data? Is there a similar AUC, whatever similar means? I like these statements. And again and again the question comes up, how similar is similar? However, the Canadian guideline, and the Canadians are very pragmatic people, as you all know, and they have give some advice. It should be 80% and more of the immediate release formulation. So if you are above 80%, you have not to argue why this is appropriate. But if it's below, then you should add all the interpretation and your arguments why this is an appropriate dosage form. And then the peak-trough fluctuation. So in other words, that the fluctuation between the maxima and minima in the profile is appropriate and not more expressed than with the immediate release formulation, because that's one of the goals for development of modified release formulations. The variability caused by the drug formulation is one of the essential points. Variability, origin for variability is various. It might be the pharmacokinetic performance of the drug substance, so drug substance-related parameters, and drug formulation-related parameters, and this has to be figured out with the studies performed with these formulations. Other factors affecting in vivo performance should be investigated, and that's very important, and I like the European guideline expressing this in particular. And then, again, the risk of unexpected release characteristics, dose dumping. So the required investigations are single-dose fasted study, and this should be done with all dose strengths. A single-dose food effect study, which is essential, done with the highest strengths, and the multiple-dose or steady-state study, again with the highest strengths. And the supposition, if there's nonlinear pharmacokinetic, the highest and the lowest strength should be investigated for multiple dosing. These are the requirements in Europe. And now the generic development for modified release forms, which is or might be most essential for you. The concept of the CPMP note for guidance is that they differentiate, and that's very important. Here, dissolution comes into play at the first stage. So there are two scenarios which are differentiated. The first one is that there are different release controlling excipients. That's the normal case, and mechanisms, but similar dissolution profiles. So they are generic and innovator product differing in composition or mechanism of drug release. However, dissolution profiles in all the media are similar. Then you should continue with an in vivo bioequivalence study, and if bioequivalence is shown, then these products may be declared as essentially similar. You know, in Europe, essential similarity concept includes the assessment of bioequivalence due to the decision of the lawyers, which are not scientists, but the European Court of Justice said bioequivalence is a parameter of essential similarity. The other scenario is that, again, the products differ in release controlling excipients and or mechanism and have different dissolution profiles. And then, under these circumstances, the normal case is that clinical studies are necessary. That's clearly stated by the guideline. And there's only an exception in rare cases, it's said by the guideline. Bioequivalence can be proven, then, of course, this is also acceptable. So let's look at the first example. That's an example on nifedipine, comparing an erosive matrix tablet on the left-hand side and an OROS system, this push-pull system, OROS or GITS, oral osmotic system, which is marketed in many countries, by the way, not in Germany, even though the innovator company is located in Germany. But anyway, here you see significant differences in dissolution performance. Here on the left-hand side, that's the erosive matrix system, which exhibits significant differences, and it's pH-dependent drug release dosage form, while the dissolution is very consistent over the entire pH range in the case of OROS system. So significant differences between the formulation. However, this was submitted to the authorities. These are the mean curves. Those of you who listened more frequently to my presentation, one of my essential statements, which I always tell my students, never trust mean curves. However, I can show you only the mean curves here, and they look pretty similar, and the conclusion of all the studies submitted to the authorities were that the products are bioequivalent. However, I have some doubts that this is really the case, because between an OROS system and an erosive matrix system, it is more or less impossible to show bioequivalence, and the profiles differ due to our knowledge how OROS systems behave. But that's another story. That's a discussion which is performed in front of the court in the Netherlands at the moment, so I will not go more into details. However, this is really unrealistic data which have been submitted. Now, what are the types of studies in case of generic development? Again, there's a single-dose fasted, the gold standard in the understanding of regulatory authorities, and they ask for the highest strengths versus the reference formulation. The food interaction study, which is mandatory, I said already, again, highest dose strength against the reference. And the multiple dose study, here you see the different scenarios in Europe where it is requested and not in the United States. Supposition for using the highest strength only is that this is a multiple unit form, a pellet formulation, and linear pharmacokinetics and those and proportional composition. In other words, they are same or identical beads used in all those strengths. If you go for single unit forms, then single dose studies have to be performed with all dose strengths because they may behave differently in terms of GI transit, for example, and that's very well established in literature. What are the requirements conducting bioequivalence studies? Again, there's a single dose fasted study as the first experiment, and there are no relevant differences compared to what we heard this morning for immediate release formulation. Total and maximum exposure are the parameters to be investigated, AUC and Cmax in other words. And in addition, there should be some characterization of the modified release properties, for example, by use of the half-value duration as a parameter. And then bioequivalence assessed in comparison to the innovator product with a full clinical dossier. The multiple dose studies, which are requested in Europe, not in the United States, and therefore the question, that I'm very interested in harmonization of the requirements between the different regions all over the world, and therefore we have to investigate more carefully the question, are multiple dose studies really needed for bioequivalence assessment in the case of the modified release products? So I have another example, and that's an example where we investigated verapamil, the innovator product, against itself, and that was using a replicate design study. And you can see here the single dose data on the left-hand side, the individual curves for one of the two products. Here you see the mean profiles, and on the right-hand side the multiple dose part, and you see much larger variability on the left-hand side compared to the situation at steady state. And you see also from the mean curves that they are a little bit closer together compared to the single dose scenario. Again, this is the innovator product tested against itself, so not different products. And the findings concerning bioequivalence were that bioequivalence could easily be assessed at steady state, and it was not possible to confirm bioequivalence here in the single dose setting due to high variability. You see the significant reduction in variability if you go from single dose to multiple dosing. So the conclusion is we should continue, and that's on our list for the UFEPS network, to investigate that a little more in details, to make recommendations based on real data and scientific recommendation to change or modify the guidelines and harmonize between Europe and the United States, for example. On the other hand, are steady-state studies useful? And here, that was announced this morning that I should talk about patches. Of course, I take up that point. And here, that is a patch example on fentanyl, and the comparison of two, the generic and the innovator product. The generic one is the black curve, and the reference, the innovator product, is the red one. And you can see from the data here, AUC and Cmax data are perfectly inside the acceptance range of 80 to 125% for both Cmax and AUC. Nevertheless, there are significant differences between both formulations. However, they are still inside the acceptance range. That's the conclusion. However, if you look a little bit more in details, we found at steady state that Cmin, which was not a parameter so far in Europe, however, the new guideline suggests to investigate that parameter as well, and I fully agree on that. However, we have to find out what is appropriate here, and therefore we will have some additional discussion. Now, here, these Cmin values at the end of the dosing interval, you know, they are applied for 72 hours, and Cmin did not meet the requirements. The point estimate was 1.33, and the confidence interval was 124 to 143%. So in other words, bioequivalence could not be assessed concerning the Cmin values. So what is the problem with fentanyl patches? In therapeutic practice, and the problems are in therapy, it is reported again and again that even though the patches are recommended for 3 days, a significant number of subjects, 1/4 of the subjects, they have more breakthrough pain on the 3rd day, and they have to change the patches already after 2 days. So in other words, the interval is too long for 25% of the patients. This is known as end-of-dose failure. That is a significant problem in long-term therapy of chronic pain, and therefore we have to investigate that and more carefully, and the subtherapeutic plasma concentrations are relevant and for that. So let's go back to the plasma profile. Concerning Cmin, the goal is that Cmin of the generic version must not fall below the affected values, and the effective values are defined by the reference formulation, because that's the formulation with all the clinical data. And you see here, that's the end of dose failure of the reference formulation, the innovator product of fentanyl, and on the 3rd day there is 40% less Cmax values compared to the 1st day. And in this particular case, the reduction in Cmax on the 3rd day is only 32% in the case of the generic product, and that's the concept, and therefore we should consider not to go for a bioequivalence approach for Cmin, but consider a non-inferiority approach, a one-sided test for Cmin, since there's no safety concern. In other words, the test product could be declared to be the better product, because the end of dose failure will be less pronounced compared to the innovator. So there's some issues for further discussion. Now, are there biowaivers possible in modified release formulations? And the answer is yes. There are exceptions defined by the guideline. For example, in case of the first modified release product, which is introduced as several dose strengths with proportional composition into the market, and in the case of linear pharmacokinetics, the multiple dose study should be done only on 1 dose level. In case of non-linear pharmacokinetics, the highest and the lowest dose strength should be investigated. In other words, some kind of bracketing may be performed. In the generic situation, several dose strengths of the generic and the innovator product, here we differentiate between single unit forms and multiple unit forms. In the case of single unit forms, as I already said, all dose strengths should be investigated single dose, multiple dose only highest strengths, and in the case of pellet formulation, also in the single dose study, only the highest dose strengths should be investigated. The supposition is, again, identical pellets or beads, proportional composition, and of course, similar, again, how similar, similar dissolution. The open issues are, you find in the guideline again and again the statement and the terminology linear pharmacokinetics, which is essential. In the existing and current CPMP note for guidance, they exchange that by the term rate of drug input, which should be linear. That is a very valid change, and I think that is more important because, for example, if there is non-linear in renal excretion, it doesn't make any difference concerning bioequivalence, because bioequivalence is focused on rate and extent of absorption, as already said earlier. Dose strengths to be tested. Here, the European position is use the most sensitive. And this is not necessarily the highest dose. It is often the highest dose. However, there are exceptions. For example, if the absorption may be limited, and then the lower dose strength might be more sensitive. So this is, again, up to you to look into the details and define the rationale for your investigation. Now, as I mentioned already, there are certain specific and special investigations mentioned by the guideline. For example, related to gastrointestinal function, there is co-administration of drugs which may affect GI physiology, and then the modified release formulation should be investigated also under these conditions. Examples are PPIs, the proton pump inhibitors, which modify the gastric pH, which may have an impact. Then prokinetic drugs, which may accelerate GI transit, or interactions with the efflux transporters and metabolic enzymes. So these are conditions which should be considered. And then secondly, drugs for patients with altered GI function, again the elderly, which are in a achlorhydric situation. So also the European become closer to the Japanese population if we become older. Then diabetic patients, of course, with gastric emptying problems, and the inflammatory bowel disease, which is also a significant problem. And points to be considered that the absorption conditions throughout the GI tract may differ, if there's an absorption window, for example, and under these circumstances, the transit of the dosage form becomes an essential point, and again, dose dumping caused by food interactions. So every agency is very worried about dose dumping, and therefore food effect studies are essential. The requirements are similar in the United States and in Europe. This is a very important requirement, and as Vinod already said, that is based on the outcome of the theophylline investigations in the 1980s. And I'd like to show you just two examples here, nicely summarized by Aziz Karim in a paper. This is the comparison of product A, fasted and fed, and you see here under fasting conditions significantly higher bioavailability compared to fed administration. However, the example on the right-hand side shows the opposite. As you can clearly see, although the active ingredient is identical, so it has something to do with the formulation. In this case, fasted state is the lower curve, and if you administer this product B under fed conditions, then you will end up with double bioavailability. Let's assume that these two products were bioequivalent in fasted state. So if you switch to fed state, one is double, the other one is only half of the values, then you have a difference of magnitude or factor of 4, and therefore it is quite obvious that food effect studies have to be performed in the case of multiple modified release formulation. However, that was the past, that were the 1980s. What about now? How is the situation nowadays? Here again an example of nifedipine once daily formulation, and you can clearly see there is a significant food effect, and that's what they normally identify as dose dumping. And this significant increase, and which is related to reflex tachycardia, and therefore the goal to reduce blood pressure cannot be achieved. So the objectives are to run a bioequivalence study of the generic versus the innovator, to identify useful information for the labeling, and also after fat administration, pH in the stomach is increased, and therefore it should be a single dose study, and multiple dose design is not acceptable here because dampens the variability. We are interested in our group in some research activities concerning dose dumping, and I always, in my little bit provocative way of asking questions, I ask the regulators again, dose dumping, is that a phantom or reality? Is that really, your interpretation really correct to understand it as a failure of the dosage form? And therefore I'd like to share within the next very few minutes some research activities. The traditional interpretation of dose dumping is that food ingredients affect the release mechanism, and the result is an uncontrolled drug release, and there's a significant safety concern. Consider the higher dose in the modified release formulation. However, there's also an option for a more physiological interpretation, and this interpretation says that the dose dumping phenomenon is caused by gastric emptying problems and not by the dosage form itself, and the release occurs in a controlled manner, however in the stomach, at a site where no absorption occurs, and then after a couple of hours gastric emptying occurs, and the rest of the dosage form together with the drug in solution is emptied into the intestine, and then very rapid absorption occurs, and this is no more controlled by drug release from the dosage form. And to show you why we came to that conclusion, I would like to share some information based on imaging studies where we follow the dosage form on its way through throughout the GI tract. We are interested in the information when gastric emptying occurs. In other words, how long does the dosage form remain in the stomach, when and where tablet disintegration occurs, and we have a very nice technique for that. That's a magnetic marker monitoring technique, and you see we have access to that building. That's in the Federal Institute in Germany, the Physikalisch-Technische Bundesanstalt in Berlin, and this is the magnetically best shielded room on Earth. The American would like to say in space, and by the way, also our colleagues from the Federal Institute, they say it's the best shielded environment in space, but I'm not sure. But anyway, in this building, which is 15 times 15 times 15 meters, there's only one room in that entire building, and that's a measuring chamber of 3 times 3 meters. Here you see my colleague Barbara Schuk under the equipment, and this is a very efficient equipment. We use QUITs, which are superconducting quantum interference devices, very, very sensitive magnet detectors. If you have a Volkswagen on the moon and start the engine, we can measure the change of the magnetic field if there would be no interfering magnetic field. So very, very sensitive, and we can follow the dosage form, which is marked by use of magnetite. Magnetite is iron oxide, which is used as a coloring component in many, many products. We need 0.1 to 1 milligram, and this is a dipole, and we put the tablet into a strong magnetic field. All the dipoles are parallelized, and then we can measure the magnetic field of the tablet. And here I would like to show you one example. That's on felodipine, extended release formulation. And again, this is like nifedipine, very rapidly and quantitatively absorbed. And the extended release formulations are manufactured to reduce the rate of drug release, since release controls the absorption, and there we would like to avoid reflex tachycardia. And on the other hand, dose dumping or food effect should not occur. So this was a crossover study, the tablets magnetically marked by use of magnetite. And here you see the mean curves. This is the mean curve in fasted state, that's in fat state. And as I mentioned already, never trust mean curves. Now I will show you the individual data of the study, which has been performed with 6 subjects. Here you see the individual curves after fasted administration, which are very, very consistent, very nice profiles, and also not too bad the situation in fat state. However, those of you, or the pharmacists, they look very carefully into the details. You will have recognized that I show you only 5 profiles. However, the study has been done with 6 subjects. And the reason why is this, what occurred here. That's some kind of dose dumping, which not only occurred here, but of course here, very significant, while the majority of the profiles were below 10 nanograms per mil. This goes up to 100 milligrams per mil. And we were interested in understanding this performance better. And therefore, I'd like to show you a little video now. Unfortunately, this will not be possible on the left-hand side, but you will see it on the right-hand side. And I think you don't need Serbian interpretation. Now, what you see here, the subject, as I have shown here, that's subject seen from the front, here from the side, because it's a 3-dimensional imaging. The red spot, that's the tablet in the stomach, and you will see it here throughout the GI tract. The time is running here, and here you see the magnetic moment which goes down. That's drug release in the inverse recording, and here you will see the plasma profiles. Now let's start the video. Hope that, whoops, why doesn't it work? Yeah, I know the reason why, because it's not my computer. So unfortunately, I cannot show it to you. I saved 2 minutes, which is helpful. But we can see, and I can explain to you what happens. This profile shows very consistent drug release. So in other words, as long as the tablet is in the stomach, up to here 240 minutes after 4 hours meal was served, until then about 75% of the drug is released and the dosage form is still in the stomach, and then it is pushed out by intake of the next meal, and then you see this significant increase, which is due to the fact that the drug has already been released and is in solution and therefore rapidly absorbed. Now, conclusions. Sorry for that mistake. I suggested myself to use my computer and then I've forgotten it. It's probably a question of the age. So what are the conclusions? The regulatory requirements are appropriate due to my understanding. It is mandatory to go for a single-dose fasted study. However, the question is it necessary to be confirmed at steady state? That's an open question, and we need more discussions on that. Food interaction studies are essential. That is for sure, and shown by the theophylline example very nicely, because food effect is not only related to drug substance properties, but often caused by the dosage form properties. And again, dose dumping, I am very sorry that I could not show you the proof why we came to that conclusion, that it is sometimes also a phantom in so far that it is not related to a failure of the product, which performs often very nicely, but it is determined by the physiology, the gastric emptying, and that is often the background for dose dumping phenomena. So thank you very much for your attention, and I'm also happy to answer more questions later. However, keep in mind, Bonn is expecting you, so please take your calendar, 14th and 15th of January. We would be happy to see you there. Thank you very much.